Bad people in the back of the room? Yes, I'm speaking to you. Josie. I don't think they can hear back there, or she's just ignoring me.
Why don't you say good people?
Good people in the back of the room, would you please come? Well, it is my enormous pleasure to be able to introduce Eva Kittay, who is very well known to many of you. Eva is a professor emerita at SUNY Stony Brook. She is one of the progenitors of the ethic of care. She has had a huge influence on my life, which started a long time ago at the Hastings Center when we did a project on prenatal genetic testing and disability. And one of the most important moments in that project was a conversation that Eva had with her son, Leo, about the meaning of prenatal genetic testing. And it just so happens that Leo is in the room today, which is absolutely wonderful. The last thing I wanted to mention is that Eva has a book coming out from Oxford in the fall, and in some ways, it's a continuation of the conversation about, in many ways, it's the continuation of the conversation about prenatal genetic testing and the meaning of disability. I've forgotten the name of the book. Learning from my daughter, the care and value of disabled minds. Wow, so I got a chance to read an early draft. I can't wait to read the published one. I encourage all of you to do. Oh, okay, enough. Eva.
Thank you very much, Eric. Yes, most of my writing about disability really started at that point. And two of the chapters of the book will be our developments straight out of our work I work at the Hastings Center. So the Hastings Center is a very important part of that book. And I am delighted to be back here and thank everyone who's been responsible for the organization, Eric and Joel. I want to say now for something different. This is a rather personal essay. As you know, I do that in my writing. And this one is almost entirely personal. No. Oh, wait a second. How do I do? Oh, okay. No. Really? My husband and I looked at each other in disbelief. We had agreed to have our daughter Sesha genetically tested for the first time in 48 years. And for the first time, we had an answer. For 48 years, we had no etiology for her many disabilities: cognitive disability, epilepsy, ataxia, hypotonia, scoliosis, and I've left out some. So, am I doing something wrong? There it is. Okay. All right. There we are. The list is, I've left out some. The list is long. There's a list of all the various ailments she has had, and I won't read them all because there are too many to read, alongside of a picture of Sasha. So I wrote here, these are 2 pictures of Sasha. Sasha is a beautiful, lovely woman of 48. She's never talked, she doesn't walk now, she is still incontinent, although with care and attention she can stay dry and clean. She does not use her hands to do much else besides grab things and put things in her mouth, and most importantly, hug. She can do nothing for herself. But she does have some receptive language, although the extent of understanding isn't clear. She has a profound appreciation of music. She loves good food, is marvelously affectionate, loves the water, enjoys musical movies. A list of what she can do, however, is relatively short. But the intensity of her engagement with those things is deep. She's been this way since birth. We only learned that something was not right at 4 months when we saw another 4-month-old baby who could lift up his head, roll over, make lots of sounds, and seemed far more advanced than Sasha. At that point, we brought her to a pediatric neurologist. The first concern was that she had some degenerative disease like Tay-Sachs. But Sasha really has not lost any capacities, she just hadn't acquired any. She was gaining weight, but slowly, nursing, but kept falling asleep, as she did. She was very floppy, could not lift up her head, was very sweet and pliant, cried very little. At 6 months old, we were told, rather brutally, that she was severely to profoundly retarded, and those were the terms, and would be so for her entire life. No one had any other labels to give us. We had a diagnosis of sorts, but no etiology, no cause. She had had a cyanotic episode, that is, she stopped breathing and was turning blue, shortly after birth, and we worried that perhaps that incident wasn't handled as it should have been. Our pediatric neurologist thought the episode was unlikely to cause so much impairment and suggested that rather than the cyanotic episode causing her disability, the reason for her disability was the cause of the episode. Sasha was also very jaundiced, as many nursing babies are, but Sasha was jaundiced for a very substantial period of time. Many years later, Sasha was already in her 30s, I was reading an article that referred to the fact that a high bilirubin count could cause cognitive disability. I had already known this, and I was only very slightly concerned that the jaundice, the high bilirubin, had caused her condition. It had never been mentioned as a potential cause, but this passage struck me hard. I found myself hysterical, screaming, crying, breastbeating, hysterical, thinking that I might have neglected her condition, and this negligence was the cause of her situation. I was persuaded by my own doctor that there was no chance at all that in the US, the bilirubin count would get high enough to cause such severe cognitive impairment, not now and not in the late 60s. I had been a careful, expectant mom, giving up cigarettes, eating well, walking lots, doing everything to make my child healthy and as good as can be. I gave birth drug-free, no drugs to cross the placenta. So while I know that feelings of guilt are possible, except for the above-mentioned bout of hysteria, I never felt guilty that I had caused her condition, nor that my body had somehow failed me. I hate even talking this way because Sasha isn't defective. She is Sasha perfect. Nonetheless, we're talking about a diagnosis, an etiology of her disability. My husband and I are both Ashkenazi Jews, and Ashkenazi Jews, like other populations who marry within their group, are prone to have genetic conditions that can be deleterious. However, genetic testing was in its infancy when we learned that Sasha was not developing normally. The only tests I knew of when we decided to have a second child, it was 1975, was Down syndrome and perhaps Tay-Sachs. We didn't really see any need for testing, and the result of that pregnancy was a healthy baby boy. Talk of genetic testing came up once again when that baby boy was to become a father. Neither he nor his wife chose to do genetic testing for any of their pregnancies. I'm the grandmother of 3. Janice McLaughlin lists a number of reasons families chose to have genetic testing for their child. Providing an explanation of why their child is not developing well. Well, no answer seemed possible. Gaining an indication of what the future holds for their child. Again, no answer was possible. Advice on use for medical treatment. None would be forthcoming if there was nothing clearly to test for. Obtaining a medical category to help in parents' attempt to receive support for their child from formal support agencies. Sasha was clinically so obviously someone who needed support services, a genetic test was superfluous. Helping them make future reproductive choices. We were unsure we wanted another child after our son, and we never thought her problem was genetic to begin with. So there never seemed to be any reason to test Sasha. Today, the techniques for genetic diagnosis have become so sophisticated that while the early techniques would not have yielded answers, now they might. What was revolutionary is whole exome testing. Now, I have to tell you, I know very little about this scientifically. I have been trying to educate myself, just to understand what it all means. So if I'm saying something wrong, just tell me. The tests do not examine entire genes. They only look at exomes, which are the part of the gene that encodes the relevant information. And at just those exomes which they know have been implicated in the phenotypes with which the person to be tested presents. These allow them to examine a limited number of exomes. And while we were unlikely to get a diagnosis before, with these new techniques, we might actually be able to do so. But what would we need it for? Well, here's the story. Sasha had developed seizures when she was about 10 or 11. We had struggled to find a medication to control her seizures and wound up through the years to try many different kinds. In addition to medications available, we tried the vaunted ketogenic diet, feeding her mostly fats and proteins to induce ketogenesis, which presumably would help with her seizure activity. This was arduous and thankless, and ultimately a futile exercise. But for a good half year, we were trying to bake our bread-loving daughter something that was a substitute made out of egg whites. Hopeless and unsatisfying. The ketogenic diet did nothing. And now we've learned that the ketogenic diet is in fact useless for people with the specific mutation that Sasha has. We tried her on barbiturates, which make her drowsier than we wanted her to be. We tried medications that only increased her seizure activity. We finally settled on a regime that was more or less effective and kept her awake mostly. We were told that her seizures were refractive seizures, seizures that could not be completely controlled. She was given a diagnosis of Lennox-Gastaut, which is really nothing but a syndrome with no explanatory value. Lennox-Gastaut syndrome simply means that she had intractable seizures and very significant intellectual delay, something we already knew without the label. At 47, her seizure activity, which had been moderately under control, broke through the medication regime she was on. From March 2017 till this past spring, Sasha was in and out of the hospital, more in than out, with bouts of status epilepticus, nonstop seizures, that is, and we were on the hunt for the medication regime that would work. We had found a new epileptologist, and he put us on a regime that so far has been quite successful, and of course. But he also advised that we do genetic testing because some diagnoses can be useful for determining what medications will be most effective. And so, at 48, we did genetic testing for Sasha for the first time. And it promised, and the reason we did it was that it promised useful advice on treatment, one of those things on McLaughlin's list. Both my husband and I were also tested. Now we were in Sasha's doctor's office hearing the results. I was certain that the results would be as unrevealing as all other efforts to learn the cause of Sasha's disabilities. I took these to be just an utterly random happening in the universe, which, as it happened, it was. But it was no longer mysterious, and it was genetic in nature. It was a random mutation. The positive result was a de novo genetic mutation on the Pura gene. That is, the mutation was not inherited from either of us. It was heterozygous. That is, it required only a mutation on one pair of the genes to be expressed. A random happening in the universe. Her mutation is known, let's see, no. There we go. Her mutation is known as a frame shift, a duplication which causes a shift in the spelling of everything after that position. So what you have there is the location, as far as I could tell, of Sasha's mutation, which means that the spelling of everything else in that gene beyond that point was not what it was supposed to be. My acquaintance, yeah, I've already said that. My acquaintance is rudimentary, but I'm going to try to convey a bit of what I've learned about the Pura gene. The Pura gene is on the long arm of the 5th chromosome. It is, no, that's going back. I'm not familiar with this kind of, all right. It is ubiquitous in living things, from bacteria to humans. It actually exists in bacteria. And it exists in all of our cells. It encodes a purine-rich element binding the protein purine alpha, which has an important function in brain development, particularly in DNA replication, in RNA transcription, and in mRNA transport. Studies in mice indicate that Pura A is required, and this is what I've got up there. Pura A is required for postnatal development of the murine brain and is involved in both neuronal proliferation and maturation of dendrites. It's been shown to be important for the transport of specific mRNA molecules to the site of translation in the hippocampus, in hippocampal neurons. Put simplistically, the protein is important in brain development in multiple ways, especially on neurons and dendrites that have particular impact on learning. So let me read from one study that describes the impact of a mutation on the PURA gene. Moderate to severe ID, intellectual disability, and neonatal onset symptoms, including hypotonia, respiratory problems, feeding difficulties, exaggerated startle response, hyposomnolence, hypothermia, gastrointestinal, ophthalmological, and endocrine problems were observed frequently. Computational analysis of photographs show a subtle facial dimorphism. Sesha has had pretty much everything on that list. That list also explains her floppiness when she was young, the fact that she didn't eat very well. I mean, it's a perfect picture. The early cyanotic episode, constant falling asleep, as epilepsy, as is the epilepsy that she developed as an 11-year-old. I'm learning that most actually develop it earlier in childhood or in infancy. The list fails to mention osteoporosis, scoliosis, and other orthopedic anomalies that make it to the other descriptions of the mutation. The early cyanotic episode, the constant falling asleep on the breast, as I said, it's all there. Oh, this is a repeat page. Receiving this news, my husband and I looked at each other startled. Our thoughts went from she has a genetic anomaly, followed shortly by this changes everything. And so nothing happened to Sesha? This was Sesha from the very beginning? Sesha was just as she meant to be, followed shortly by, well, this really doesn't change a lot. It was also strange. We were certain that something had happened to Sesha along the way. But there was no Ur-Sesha that was to grow into, and I hate the word, so scarce grows around normal, into a normal woman. There was no longer a mysterious narrative of Sesha becoming disabled. Sesha, with all her disabilities and all her charms, was just Sesha through and through. A brief feeling of uncanniness coursed through me, and a brief moment of loss. I could understand an Ur-Sesha to whom something had happened, but it was harder somehow to wrap my mind around this thought. There was never some normal Sesha. Without her disabilities, she truly never would be Sesha. That thought actually had never occurred to me, strange as that might sound. In a moment, the thought of which I'm really excavating now as I speak, as I write, that there was once more, now after 48 years, a loss, though very momentary. Not the loss of the daughter I had expected to have but didn't, but a loss of a normal daughter to whom something had happened. I had to adjust to the idea that Sesha was just Sesha, or as the parents of the Pura group, parent Pura group say, she was always Pura perfect. And yes, as rare as Pura is, there's only about 200, a little over 250 that are diagnosed to date in the world. But this was only discovered in 2014. So it's very possible that a lot of people simply haven't been diagnosed. So we don't know the true number. As Jeffrey and I moved from this changes everything to but it also changes nothing. It was clear to us that Sesha is still the daughter she always was. She's still the person she always was. She is still beautiful, lovely, sweet Sesha, whom we utterly adore, and with whom we fell in love the moment we saw her. As uncomfortable as it is to say, to voice, it took me a few seconds to get over that sense of strangeness, a strangeness that would momentarily, fleetingly pass through my mind for several weeks after the diagnosis. There was a way I needed to reconnect with Sesha, although, of course, I didn't at all. On an emotional level, there was no rupture whatsoever. But on another level, which I can't quite call intellectual, and surely can't call emotional, but perhaps on some intuitive or reactive plane, there was a slippage which I needed to monitor and restore to its previous equilibrium. Moreover, I was, very slightly but still, concerned that I might have to change something in my writings defending Sesha's personhood. And I've done a lot of writing defending the personhood of people like my daughter. I take personhood to be relational in at least two ways. She is in relation to me as my daughter, and she stands in the same relation to all other humans as do any of us. Would the fact that her impairments were a consequence of a genetic mutation pose a problem to my thesis? Is she any less a daughter of mine because of the genetic glitch? Is she any less human because of a rupture in an identity that comes from the genetic code? No, she is no less a daughter of mine genetically. She has all the genetic material, both the duplication in one spot on the long arm of chromosome 5. No, she is no less human either. Mutations are a part of our evolutionary history. If no humans had mutations, we would doubtless have died out before we ever got to be the humans we are. Yes, Sesha had a mutation, but aren't we all mutants? I had just completed reading Peter Godfrey Smith's fascinating book, Other Minds, the Octopus, and the Sea, and Deep Origins of Consciousness. And he explains the source of senescence and death. These are a feature of an evolutionary process. An organ's vulnerability to prey or other external forces that might kill it helps to determine the age at which it will procreate. The more vulnerable it is to prey, the earlier it procreates. Furthermore, evolution is made possible by the inevitable mutations that occur as DNA is copied in the process of reproduction. These mutations are the driving force of evolution. Some mutations are beneficial to the organism and can help it adapt to changing environments, but most are deleterious. And in that most mutations impede our ability to survive. Bad monogenetic mutations that occur early in life, such as Sesha's, are rare. Most of those are eliminated in the process of natural selection. But there are also polygenic mutations, and at least two genes are involved, and both need to have the mutation if a phenotype is to be expressed. Among one of these mutations has a beneficial effect when people are young, but the other is necessary to enable that beneficial mutation to be expressed. That other one turns out to be deleterious when we grow old. But as the deleterious ones are coupled with the good effects when we're young, they are not weeded out in the gene pool. They remain and cause problems after our procreative years. As populations evolve, these grow more prolific. Most of us will die, he explains, because of some deleterious mutation which thanks to it being part of a polygenic mutation, have remained in the gene pool. My dearest and oldest friend had, while Sesha was in and out of the hospital, died of just such a cancer-causing mutation, one that, as it happened, was de novo. My dearest friend was a mutant, as was my daughter, as are we all. There are between 100 to 200 new mutations on average in each person. These are so-called errors in the copying over of the genetic material from each of our parents. In The Gene, An Intimate History, Siddhartha Mukherjee says, "We have seen the mutants, and they are us." He cites Archibald Garrett, an English pathologist, saying, "Genes make us human, and mutations make us different." If you're learning that your child has a mutation, is it better or worse to learn that the mutation was de novo? Janice McLaughlin, along with her various co-authors in a series of papers, studied the effects of genetic testing on children, on the families and their perceptions of themselves as a family. She notes that parents often blame themselves and feel responsible for passing down genetic inheritance that has a negative effect on their child. She goes on to say that geneticists do need to perform a kind of moral labor when communicating a de novo finding. They can stress that it means that parents have no reason to blame themselves, either for passing this on or doing anything in the pregnancy that would have contributed to the problem. On the other hand, my son's sitting in the audience, I will say this. As we were talking about this, he said something that had just occurred to me right before, and that is that, on the other hand, the sharing of the genetic trait can have a positive effect. One young girl was told she had the same genetic trait as her mother, which was the reason both had learning disabilities, and she explained to us that this made her happy because it meant they were the same. As I never felt particularly guilty in the first place, a de novo diagnosis did little to relieve any non-existent worry. However, weeks after the diagnosis, it occurred to me that an inherited mutation would have constituted a connection that a de novo mutation denied me. If the condition is inherited, the apple that has fallen far from the tree turns out to be not so very far from the tree after all. Erica Haynes, who studied families where a child was tested, remarks, "Children who are seen as somehow different have to have a space found for them in order to locate them within the web of family relations." Yet we didn't need to locate Sesha within our web of family relations. Despite this jarring bit of information, Sesha was firmly in our web of family relations. If anything, she stood at the center, very much making us all cohere as few families cohere. Many have said to us, "You must feel so relieved to have finally found out the reason for Sesha's disabilities." And I must confess that this always strikes me as odd. It's no relief except to know that her impairments were not caused by anyone's negligence, and perhaps my son and his family to know that they have no more chance of having a child with Sesha's condition than any other person on earth. Otherwise, it hardly matters that we now have an etiology. Had it helped us pinpoint a useful seizure medication, it would have been a help rather than a relief. Unfortunately, the diagnosis has not helped in that way, although it might in the future. It did inform us that the misery we all endured trying the ketogenic diet was for naught. For parents who have a sense of guilt about the disabilities of their children, the genetic diagnosis would be a relief, but that's not our story. It did, however, and for this I'm very grateful, put us in touch with parents of other children with Pura. I learned that Sesha is by far the oldest of those diagnosed, and it was very exciting for the parents to learn that 29 was not inherently the upper limit for Pura children. That was the only child they had. The oldest one they had was 29 years old. And here they find out that there's someone who's 48 who has the same genetic mutation. It was rather uncanny to see the pictures of these children. So many reminded me of Sesha when she was young, the facial expressions, the twist in their bodies, the ataxic walk. And for some reason, the facial dysmorphia does something to enhance the beauty of these children. I expect that this network of parents and network of researchers that I have connected with may prove fruitful in the future, if not for Sesha, then for others with this mutation. It is the discovery of what Andrew Solomon called a horizontal identity for Sesha and for us parents of children with Pura mutation that is perhaps the most transformative aspect of the diagnosis. I'm running out of time. I did want to, it's kind of late in the game when I did find this, okay. Maybe I have it later, so I'll just ignore that. All right. I've identified 4 ways in which Sesha's diagnosis roiled the waters. I can characterize these as the loss of an Sesha, who was not disabled, the otherness of a de novo mutation, and a sense of loss of connection with your child. The first of these losses is rectified in realizing that the duplication of the string AGCCG and the framework shift that are responsible for Sesha's impairments are really not any different in substance than had she suffered an oxygen loss at birth. She is my genetic offspring in exactly the same way. A tiny bit of the copying mechanism got mixed up. Mistakes are inevitable in all organic life, and nature makes them too. Spelling mistakes, no less. That's the nature of nature. The full force of those spelling mistakes I've lived with for 48 years, and yet I still am not sure of what Sesha knows and understands. Sesha herself is neither sheer mystery, nor is she's not sheer mystery, but she's still mystery. Nor is she a mistake. She is the person she is. She's no less human, no less my child, no less a member of our family, no less an equal member of the moral universe. Whatever imaginary Sesha I lost when I discovered I landed in Holland, I'm referring to an essay I'm assuming most people know, and not in Italy, that imaginary Sesha is sitting somewhere on an Italian isle. But she is not my daughter. My daughter is in Holland. And if I'm to be a good biocitizen anywhere in the world, I claim my citizenship there, for it holds this precious inhabitant and all these other faces of Pura children. That's it. Thank you. Questions? This is a deflationary talk, right? I mean, we have this incredibly sophisticated science, a science that's so sophisticated it wasn't even in existence, 10 years, even 10 years ago, right? What difference does it make?
Thank you so much, Eva, for that. As always, beautiful talk. This was a different talk, but a beautiful one as so many. I thought I would just share with you a line that Millie Solomon spoke to me yesterday after Catherine's talk. Is Catherine? Hey, Catherine. Millie said maybe we should have titled the conference "The Gift, the Weight, and the sometimes irrelevance of genomics.
Right. Yes, I'd be that third circle. But you're going to have another metaphor besides binocular thinking then. It's not to say that it couldn't be relevant. I mean, it could have, yeah, I mean, I keep thinking about this. I mean, it could have, and it might still give us a more precise medication, right, for Sasha's disability. And if it did that, I would be quite grateful. It does have a certain social impact that, together with, social media, that parents from every part of the world can be in touch with one another. And for some people with some syndromes, that's been very helpful. Since I'm so far along in the game, and there's nobody else who's as far along in the game as I am, it's proved less helpful for me. But again, it might still prove helpful. But what struck me really is this idea that it should be such a relief to me to finally know. And it really isn't. It took this kind of momentary strangeness suddenly, Sasha wasn't quite who I thought she was, but she is who I thought she was, The other thing, and I sort of wanted a postscript on this, is what would have happened if we had all this information before I had Sasha? And I confess, I would have aborted. I mean, look what they're telling me. She's going to be she's going to be profoundly retarded, right? She's not going to be able to walk, talk, because most of the kids neither walk nor talk. Be incontinent all the rest of her life. Because you get all the string of negativity. You don't get the positivity of this individual person, right? So, would I have said, oh, I'm taking it all on board, whatever? Probably not. Probably not. And that's actually quite profound. Yes, Jenny?
Just following up on that last point, I'm curious how today this information is being presented to potential parents, and whether or not it is being tested for. And I don't know, there's certain tests that are put forward as, it's on the panel, everyone's offered it, and I don't know where things are at with this particular variant. You're saying it was only discovered in 2014. So I'm both interested in what you know about that, and also how you feel about that, and to what extent we're talking about citizenship, and you're talking about, your feelings as a mother, but also as a member of a community. I'm wondering what kind of, sense of obligation you've had to influence how policy develops on these types of matters.
I don't know if this is offered. It is offered?
Yes, but not much, I think is the answer. So if you do amniocentesis or CVS in some places like New York, well, in one clinic here, they are doing on a sort of experimental basis whole genome sequencing on samples. That's not what is always offered now, but that would potentially pick up this kind of pinpoint mutation. But if you did CMA, it wouldn't, I think. So, which is more standard of care. So there's a move to do whole genome sequencing on fetal samples, and there's also a move. So we have a project on the future of prenatal testing. We'll be writing more about this very soon, but and that there is also a move to do non-invasive whole fetal cell sampling, which would allow whole genome sequencing on fetal cells that were taken non-invasively from a cervical smear or from a blood test. So there will be, I think, a technology in the future where non-invasively at like 8 to 10 weeks gestation you could do whole genome sequencing, and then that raises all those questions about what is sort of routinely looked for or returned, however you want to. That's why I'm so attuned to this whole we found it, so we have to give it back kind of narrative, there is basically absolutely no way in which we're prepared to give this kind of complex information to 4 million to 6 million women a year in the US alone, right? That's how many pregnancies there are. So and then, like you were just saying, there are those two very hard to hold together notions of what the medical description would be of the condition, which could vary greatly, but would be an exhaustive description that was pretty frightening to encounter. And some non-invasive prenatal tests screen for things right now that have that kind of scary list. So microdeletions and duplications look like pretty much like that list. And then the reality of families who are joined in the way that the Pura family is and other conditions like that, where there is huge celebration and joy and like kind of togetherness of those families and their children. And it's like, how do you, and you know, the Down syndrome history is just a perfect example of those tensions which people are navigating, but it's complicated, and it's complicated to do it for like 1,000 conditions.
Yeah, and you know, the level of expressibility and penetration. There are some of these kids who apparently are able to read. There is expressive language. Whether all the disabilities are profound, I don't know. But, you know, it's so interesting when I read the parent groups, the way in which they describe their children is so much the way I describe Sasha. Is the genetic mutation connected to her sweetness?
I'll just chime in for a second here as the brother. I hadn't thought I hadn't heard this yet, so it was great. It was a big deal to me. It was and I'm trying to sort of figure out exactly why. I think certainly when we had our kids and we thought a lot about genetic testing, it's something I thought a lot about before even thinking of having kids. The fact that I was sort of going on the presumption that what Sasha had wouldn't have come up in a test, and that was something, as we thought of the pros and cons.
It wouldn't, by the way.
And it wouldn't have, right. Although it may someday. So going on that was sort of useful in weighing the pros and cons for me, because the thought was, well, look, if what Sasha had didn't come up, why are we sort of going down this road? It's just, you know, it's such partial information that is it worth the sort of the cons? But learning about it was a huge relief, because my kids want to have kids, and not having to deal with this aspect of that with them is important, and a big weight off of my wife and, you know, sort of our minds, and, you know, your minds too in that way. I guess the other thing I'd say is that, I guess just personally for me, learning, you know, one thing about having Sasha as a sister is that it's very, the unknowns are very kind of dizzying. Why she is the way she is, how is she really, what does she understand, how do I communicate that to others, who's going to get it, who's not, and was a big part of my life. And there's something about learning that it's all because of a single genetic anomaly that makes it less dizzying, that makes it more, I'm more oriented in it. I certainly don't understand her any better, but the sort of list of unknowns and the list of, sort of the number of shrugs I have to give when talking about her is less, and that's empowering in some ways. And I also think that there's something about a genetic anomaly that's more relatable. When explaining Sasha, when introducing Sasha, that everyone sort of knows a little bit about what a genetic anomaly means, whereas not even knowing if it's a genetic anomaly or the result of something that happened or, some spark from God is all a little, again, kind of dizzying. So the idea that it didn't really do much to change is, I get your point and you're right. In the day-to-day, it doesn't mean, if we hold her up this way, she'll walk better. It didn't give us sort of things like that. But it does have some less tangible benefits.
Thank you. Thank you for telling me that. See, we need to have this forum to have a discussion about these things.
I think this is the last question.
I was just reflecting on this idea of kind of knowing and not knowing, and how did we know and how did it change? And it makes me think about kind of the whole process of epigenetics. And whenever I read some new epigenetic study, I'm often struck by, but social scientists already knew that. So some study comes out and shows that if you're the victim of domestic violence during pregnancy, that affects the epigenetic programming of how sensitive the cortisol, response is in the child, and then they're more stressed. But social workers already knew that if, you were, the victim of domestic abuse during pregnancy, your children exhibited higher stress symptoms or behaviours. And so just the kind of this, I feel like that your story is kind of indicative for a lot of what we're seeing in genomics and epigenetics, how much of it is new knowledge and how much does it sort of change what we should already be doing anyway. Yeah.